going to go over the simplest of stains in your time here in microbiology lab, which is actually called the simple stain. So in this technique, we're going to be using a solid culture, a slide, and a couple of staining reagents that will help us visualize bacteria better under the microscope. The first step in this process is going to be sterilizing your loop. You guys already know this technique from learning aseptic technique in the last lab. So we're going to go until it gets red hot. Let this cool for approximately 10 seconds. And then what we're going to do is place 1 drop of water. What we're going to do is squirt a little bit of our distilled water onto our disinfected bench, pick up 1 loopful of water, and put a little dot on our slide. This will help spread the bacteria out better when we go into our solid culture. So we sterilize again, let the loop cool, and then we are going to retrieve about 1 colony. So on this plate you can see there are many isolated colonies, so we're going to pick just 1 tiny colony, scrape it up with our loop, put this back down, and we're going to spread it out with the water so it dilutes our sample. This will make the individual bacteria easier to see with our simple stain. Then we sterilize the loop 1 last time. Now what we have to do is let this dry. So we're going to take our clip and we're going to very carefully place it over the top of the flame just to help the water evaporate a little quicker than normal. This will also heat fix the bacteria so they stick onto the slide better and don't slide off when we put the staining reagents on them. The next portion is to stain our slide and our bacteria. So with a simple stain, you can actually use 1 of 2 reagents. You can use crystal violet, which will make the bacteria violet, purple, as it suggests, or you can use saffronin, which will make them look pink or red. Today in this demo, we're going to use crystal violet. So with these little tubes, they're kind of finicky, but you want the top to line up with this little thing on the side. This will allow some of the dye to flow onto your slide. So we're going to place it over. For real? Ew, it's a little crusty. And we're going to just cover our sample. Just like that, not too much. Now we're going to let this sit for approximately 30 seconds. Now, after our dye has been on our sample for 30 to 60 seconds, we're going to rinse with just plain distilled water until it runs off the slide clear. So we're going to start at the top, and we're going to aim our slide into the dish, and we're just going to gently rinse all the excess crystal violet off. Make sure you get the back as well. See how the water is now running off the slide relatively clear? That's when you know you're ready to go. So you can actually see your sample that it's stained on your slide. Next, we're going to use what's called bibulous paper, or just plain blotting paper. We're just going to place our slide in between a couple of sheets, and we're going to gently blot, like the name suggests, until dry. The last part is going to be observing our sample under the microscope. So we're going to place our sample right on the stage. We're going to focus it over an area where there's a good amount of stain, but not too much where it might be too thick to see. And then we can observe under any of these lenses. Don't forget, you can't use oil with anything but the oil lens, and make sure to clean right when you're finished.